tackling COVID-19 pandemic, current scenario and upcoming developments, we have a guest of honor with us, Professor Viney Jain sir, who is former Director of Institute of Nuclear Medicine and Allied Sciences, Defense Research and Development Organization, New Delhi. I heartily welcome you, sir.
Thank you.
सर, ये हमारा बहुत बड़ा सौभाग्य है कि आज आप हमारे बीच हैं और हमारे साथ अपने विचार साझा करने के लिए यहाँ उपस्थित हैं। बहुत बहुत धन्यवाद सर। We also have Director of Morarji Desai National Institute of Yoga, Dr. IV Basavaraddi sir with us. Welcome, sir.
Namaste, namaste.
We also have Communication and Documentation Officer of MDNIY, Mohammad Tayyab Alam sir with us. Welcome, sir.
Thank you, Vandana.
अब संस्थान की परंपरा के अनुसार प्रार्थना से कार्यक्रम की शुरुआत करते हैं। ओम सहनावतु सहनौ भुनक्तु सह वीर्यं करवावहै तेजस्विनावधीतमस्तु मा विद्विषावहै। ओम शांति शांति शांति। Now I would like to request Communication and Documentation Officer of Morarji Desai National Institute of Yoga, Mohammad Tayyab Alam sir, to deliver welcome address. Sir.
Thank you, Dr. Vandana. Good evening, everyone. It's my pleasure to invite you all in the Friday webinar wherein Morarji Desai Institute of Yoga, the Ministry of Ayush, Government of India, organizes a lecture on every Friday by inviting eminent experts of modern medicine, yoga, allied sciences, media, diet, and etc. Today's lecture on tackling COVID-19 pandemic, current scenario and upcoming development is being delivered by one of the eminent scientists of modern medicine. Today we have with us respected Professor Viney Jain sir, former Director INMAS, DRDO, Defense Research and Development Organization. Professor Viney Jain sir is one of the prominent biophysicists and radiation biologist both in India and abroad. We are extremely thankful, sir, for accepting our invitation in a very short period of time. On behalf of Morarji Desai Institute of Yoga and my own behalf, it's my privilege to welcome you, sir, on our Friday webinar, which is hugely popular in social media platforms, sir. Welcome you, sir.
Thank you.
I also extend my warm welcome to Dr. Ishwar V. Basavaraddi sir, Director Morarji Desai National Institute of Yoga, inviting eminent experts of yoga and other fields on every Friday is becoming possible only due to the untiring efforts of Director sir. You also welcome, sir, in our Friday webinar.
Thank you.
I also welcome all the staff, student, social media team of the institute and social media friends who are joining us from India and abroad. Thank you.
Thank you, sir. Now I would like to request Director sir to deliver introductory remarks. Sir.
Om Shanta Shanta Shanti. Respected Professor Jain Saab, Shri Tayyab Alam ji, Dr. Vandana, my colleagues in the institute, student friends and yoga enthusiasts who are joining this weekly webinar from different social media platforms of the institute across the globe. Hearty welcome to you all. Today I am really very happy because there are several research institutes national institutes under the DRDO like DPAS and INMAS and others. And I had a very good colleagues and we worked together for many of yoga related projects. Today we are very happy to have Professor Viney Jain ji, who is the former director of Institute of Nuclear Medicine and Allied Sciences, DRDO, New Delhi. As said by my colleagues, Professor Jain is a biophysicist and a radiation biologist, and he has received his postgraduate and postgraduate doctoral training from the very prestigious universities in the Europe and America, especially in Frankfurt, Germany. He has been the recipient of the fellowship of the German Academic Exchange Service and the prestigious Research Fellowship Award of the Alexander von Humboldt Foundation, Germany. He is the author of more than 150 original scientific research publications in peer-reviewed index journals. He served on the faculties of the Physical Research Laboratory, Ahmedabad, All India Institute of Medical Sciences, New Delhi, National Institute of Mental Health and Neurosciences, Bangalore, NIMHANS, at Delhi University, and as a visiting professor and scientist at several universities in India, Germany, UK, France, Netherlands, and USA. Professor Viney Jain is known for his original research contribution in developing different modulators of radiation responses and their applications in improving cancer therapy. Besides biophysics, bioenergetics, and oncology, Professor Jain's interest includes philosophy of science, development of eco-friendly living, and religion, spirituality, science dialogue. His current studies are focused on leisure, dhyana or meditation, forgiveness, and behavioral modification. So, from scientist, slowly, with his experience, he is converting into a spiritual scientist and a man of yogi. So, from such a learned scholar, he is talking on the topic tackling COVID-19 pandemic, current scenario, and upcoming developments. In his lecture, he will present and discuss a holistic approach in the management of COVID-19 pandemic, integrating some of the new advances such as use of 2DG in modern medicine, Ayurveda, yoga, and also meditation as independent practice. He will also deal with aspects of prevention and also management of COVID pandemic. The emphasis should be on the application of simple, feasible, and cost-effective technologies which are the need of the hour. At this juncture, it's my proud privilege to welcome Professor Jain Saab. We are honored, sir, with your esteemed presence, and thousands of people will join this webinar. Some people will join on live, and some people will with our YouTube, etc. Throughout the week, many people will be very fortunate to listen to your scholarly lecture. Just before I hand over the mic stage to you, sir, our institute from the last 6 months conducting this more than 7 months. We have started on 2nd of October on Gandhi Jayanti, and many eminent scholars like you has participated. This has become a very popular lecture series, and the very compact format of the lecture will put on the ministry's social media platform for the dissemination of the knowledge given by the eminent scholars to the general public, where lakhs and people are the beneficiaries. So our institute is doing a trying our level best to invite scientific community, yoga professionals, philosophers, spiritual leaders, social workers, journalists, and take their knowledge and serving to the general population at large. With these few words, once again, it's my privilege to welcome you, sir. Hearty welcome. And I request you to uh, deliver your talk on tackling COVID-19 pandemic. Professor Jain sir.
Om Arham Namah. Jai Jinendra. Pranam. Salam Alaikum. ये मेरा एक सौभाग्य है कि आप लोगों ने मेरे को ये अवसर प्रदान किया. So I am very grateful to the staff, faculty, students and Director Sri Basavaraddi Professor for arranging this, for giving me this opportunity. Now, I would like to first of all pay my respects to my teachers and students and all the workers who contributed to these developments, that today we have 2-deoxyglucose available for treatment to the patients. And I'm very happy that so many years of work, it's more than 50 years of my work, has helped in the development of this 2DG as a possible treatment or as a potential treatment for COVID and for cancer therapy. Now, since I have a lot of material, I have tried to put in a few slides, and without wasting any more time, I'd like to come to the topic. Can I share these slides, please? The COVID-19 pandemic is created havoc throughout the world. And in India today, as we all know, the phase 2 is ruining the lives of so many people and creating a lot of mental stress and pain in the general public, in the families, and to the patients who are being diagnosed as having a COVID-19 positive. Therefore, the present scenario is really a lot of information is available on the TV, electronic media, print media, and social media. And I will simply show you some of the major problems which we are facing in the current scenario, and then come to the upcoming developments. नहीं, इससे पहले हुआ ना, वो नहीं है. Well, in the current scenario, some of the major difficulties, as you know, is the availability of hospital beds, then lack of professional skilled health workers, and lack of oxygen, lack of non-availability of medicines. Actually, no effective medicine has been discovered so far, but even those which have shown some benefit, they are not available easily in the market, or they are very expensive. And another complexity which people have talking about today is the infection, fungus infection in the patients who are suffering and who are under treatment in the hospitals. A number of deaths have been reported in the last week. So another big problem is the mental stress and anxiety and fear of this among the public. And therefore, this also has to be tackled currently and in the post COVID era. So we must take a holistic view of all things, as Dr. Basavaraddi has told you in the introduction. And that's what I'll try to do and give you some ideas about this. So first of all, I'll come to the therapeutic energy nutrient modulation by 2-deoxyglucose, 2-DG. Then I may briefly talk about some prevention of fungus infection and acute oxygen need by means of methylene blue. And then the role of alternative medicine, and including Ayurveda and homeopathy. But I will mainly focus on the 2DG applications and on the application of yoga and meditation for removing the mental disorders and behavioral disorders. Because one of the problems has been also that in the public, people have not been following the appropriate guidelines for COVID. And they are not wearing masks, or they are not following social distancing. And therefore, the problems are becoming more difficult to tackle. And this has to be also tackled and for all times to come. And the behavior modification is very important, but it's not easy and it will take time. But we must make an attempt to start these things in a big way. So next, I'll talk about first of all, about the 2DG. And I will give you briefly a historical background, rationale and concept, development, mechanisms of action of 2DG, what we understand today, and what are the questions which are being asked, and what can be the future directions. Now, the historical background of developments in India. Now, before this, the international development of 2D, I'll say a few words. Actually, this is about 80 years old, that the work on 2DG started in USA. Sorry. And the work first of all was carried out in East, and later on it culminated in the clinical trials being carried out in NIH in Bethesda. And they tried 2-deoxyglucose alone and found that that was in cancer patients, and found that a single dose was not effective. They gave intravenously. And when they tried to give every day or alternate days, then they found that the 2-deoxyglucose was very toxic to the patients, although the tumors didn't grow, but the patients could not tolerate it. And after this, the work on 2-deoxyglucose for cancer, and especially in the clinical applications, suffered and was discontinued or slowed down. At that time, I was working in 70s in University of Frankfurt, and I finished my PhD in 1970, working on yeast and studying the energy relationship of growth. And when I came back to India to AIMS as assistant professor in Department of Biophysics, then we continued this work within the framework of Indo-German collaboration, which was decided upon by the Indo-German collaboration by the two governments. And Director AIMS, Professor Ramlimka Swami at that time, and my head of the department, Professor R.K. Mishra, they encouraged me and were instrumental in making this Indo-German collaboration possible. So we continued some work, basic research on yeast cells, and also we developed some other models like the mammalian cell cultures and the mouse models, and tried to study the effects of this as preclinical work. The work on mice, murine, normal mouse and tumor-bearing mouse showed two very important things. One is that the whole body radiation of mice with gamma rays, when given 2 dg, it reduced the radiation damage and the mice survived longer. Second thing, in the murine tumor models, which was carried out in Germany in the Institute of Biophysics and in the German Cancer Research Center in Jülich, and with the help of Professor Feinhagen, we showed that in the tumor-bearing mice, the 2 dg was beneficial in tumor, and most of the effects were in the hypoxic subpopulation of the tumor. This was possible to do with the interesting technique of radio labeling the tumor cells with the iodine 123 and iodine 131, and then following these activities. It was a very nice technique developed by Professor Feinhagen. So this work on model systems in AIMS and in Germany led us, encouraged us to try it out in the clinical trials on human cancer patients. And the first clinical trial was done in AIMS on thyroid carcinoma patients. Professor A.K. Basu was the head of the nuclear medicine department, did this work, and then presented also in the Department for Atomic Energy and the Conference of Nuclear Medicine. But then I was invited to join NIMHANS in Bangalore as professor and head of biophysics, and therefore the work which we were carrying out in AIMS that slowed down. So in NIMHANS, Professor GNR Reddy, who was then the director, he encouraged me, and with the help of neurosurgery department and with the help of radiotherapy department in Kidwai Institute of Oncology, we carried out some work with the help of my PhD students. And I must especially mention the name of Dr. B.S. Dwarkanath, who developed new techniques in organ cultures of biopsies from the cancer patients, first of all, from the brain tumor cancer patients. And he did some in vitro work and got some very encouraging results. And this encouraged us then to initiate some clinical trials. And Kidwai Institute of Oncology, the director, Dr. Bhargava, and later Dr. Ananta, they were very helpful, and they started the clinical trial. And Dr. Mohanty, B.K. Mohanty, he did the first clinical trial on GBM patients, brain tumor patients, in 1986, actually, or beginning of 1987. But then I was invited by Dr. Arunachalam, who was then the scientific advisor to Defense Ministry, to join as director in Institute of Nuclear Medicine and Allied Sciences, because General Lakshmipati, who was the director, he was retiring. And I accepted this invitation after some time. And so in 87, I joined there. Then the collaboration between Kidwai Institute, INMAS, NIMHANS, and AIMS, that continued further with the help of Indo-German collaborative project. And next. ये नहीं होगा पहले So in mass we organized a seminar on this 2-deoxyglucose and its role in the radiation oncology, and where very prominent radiation oncologists and radiation biologists from both the countries joined. And these proceedings were then published in 1990 by ICMR. And this seminar was funded by ICMR, DRDO, and the GSF, that is Council of Radiation and Environmental Research, which was the organizing partner in life sciences for the Indo-German collaboration. Next. Now I come to, with this brief background, I come to the rationale of 2DG for COVID-19 treatment. Now, once the SARS-CoV-2 virus, or any virus for that matter, gets an entry in this human system, then the virus infected cells, they go a metabolic reprogramming. And this reprogramming is similar to what happens in the cancer cells. And since we had very good results with the cancer cells, we thought that this could also be tried to treat the COVID-19 patients. But later on, we searched the literature and we found that actually this idea was very old. In 1970s in the USA and Europe, some people have tried this in vitro and found that 2-deoxyglucose was very effective in inhibiting the growth of the virus. And they tried in various kinds of viruses, and for herpes virus, they got also a patent in USA. So we were encouraged then, after seeing all this literature, that our suggestion of using 2-deoxyglucose in the virus infections is valid, and we thought that it immediately some clinical trials should be taken up here. And this work was then supported by the then Director of INMAS, and also then they discussed this with Dr. Chandna and Dr. Bhatt, and then a proposal was sent to ICMR for giving permission for the trial. Now, why this 2DG is effective? Now, first of all, what we think is that after the entry itself of the virus, which happens through the spike glycoprotein binding to the viral receptors on the cell surface, this binding, this entry is also inhibited by 2-deoxyglucose. Now, this work was done by the group of Dr. R.K. Sharma, who was my student, and then after retirement joined as Vice Chancellor in the Savita University in Chennai. So I spoke to him also, but then he said that we are not yet equipped for viral work in our laboratories or in medical college, but I can do a theoretical study because his student, Dr. Parul Thakur, Pallavi Thakur, she was working at that time in the Patanjali Research Institute in Haridwar. So he spoke to Parul, and she agreed after getting permission from the director there, that is Acharya Balakrishna. So this is how it has started, and this work was completed and now is under publication in the annals of National Medical Academy, and will appear perhaps in a few weeks. So, but we put the preprint on the ResearchGate, and it became very popular. Lots of people started reading it. But this was a theoretical work, and we have to verify whether the entry of the virus in the human cells is really inhibited by 2-deoxyglucose. If it does, it will have lots of implications. But after entry of the virus, the programming of the host cell metabolism happens, and as I told you, it's like the tumors. And the demand of energy by the virus infected cells increases, and this has to be met for the viral replication. Next. So now I come to the basic idea of using 2-deoxyglucose or energy deficient diets or other procedures for this. Now, one important thing one has to remember that in the living systems, the energy yielding processes and the energy consuming processes are very tightly coupled. And this is a very efficient use of energy and nutrient resources in the living systems, because during evolution this has become a very, very efficient process. But under certain conditions, this imbalance between demand and supply develops. For example, in the tumor growth, or in the virus replication, or diabetes and some other diseases, this imbalance occurs. Now, we have to restore this balance. Now, what are the methods and benefits of controlling this energy demand and supply? See, we know from our old ancient practices that regulation of breathing and regulation of blood flow can be done by means of pranayam and by means of yoga. And second important thing is the regulation of food by nutrients and intake. What kind of food we take, vegetarian or non-vegetarian, high energy rich food or high protein rich food. So this regulation of food by for nutrient uptake is very important. Type of and pattern of diet, when do you take, very important. Role of fasting has to be investigated. And the metabolic engineering, that is how we can affect the whole metabolism. This is becoming a very big field in today's medicine. This has to be investigated and understood. So if you do that with the regulation of energy metabolism, then there have been lots of benefits, like the self-control, emotional control, reduced stress, improving quality of life, therapy, and longevity. In NIH in Bethesda Institute of Geriatric Research, they have shown that when 2-deoxyglucose is given in the diet orally for a long time in monkeys, then the lifespan of the animals increased. And also, if there is a calorie restriction in the diet, it leads to longevity. Next. So, I will now come to the viral infected cells and cancer which need a high energy flux, as I told you. Now, the energy in living system, as most all of you know, is obtained by two processes. One is the glycolysis, another is the oxidative phosphorylation linked to respiration. The two systems of these major systems of energy production, they are usually interacting and balancing each other. But in case of high energy demand, it has been observed that the glycolysis and the glucose play a major role, and the oxidative phosphorylation and oxygen and mitochondria production in mitochondria, this is suppressed, and this effect was observed by Otto Warburg in Germany. Otto Warburg in Germany, who was a Nobel laureate, and he already published this in 1924, über den Stoffwechsel der Tumoren, the metabolism of tumors. And he observed that the aerobic glycolysis in many tumors is suppressed and anaerobic glycolysis is increased. And this was called the Warburg effect. And it is has been found now that it is one of the fundamental change in cancer cells, in most of the cancer cells. It's valid. So lots of research is now going on this effect. Next slide. So how this 2DG in, selectively reduces energy supply in tumors. 2-deoxyglucose is a molecule which is very similar to glucose, only in carbon number 2, the OH is replaced by H, the oxidation oxygen. So that's why 2-deoxyglucose. Now, this 2-deoxyglucose enters the cell just like glucose, because the glucose receptors on the cell surface, they are not able to differentiate between 2-deoxyglucose and glucose. But once it enters the cell, then it is phosphorylated to 2-deoxyglucose 6-phosphate by glycohexokinase. And 2-deoxyglucose 6-phosphate is not able to be metabolized further. It inhibits the further glycolysis and the pentose phosphate pathway, PPP pathway, and therefore it inhibits the growth of cells. Next. Now we have studied this in very many model systems, including the mammalian cells, and our major idea was, with the 2-deoxyglucose, that if you combine it with cytotoxic agents like radiation or chemotherapeutic agents which act on the DNA, then what happens is the 2-deoxyglucose is able to inhibit the repair of these damaged cells. And for this, energy is needed. Is pointer right? So what we found is that the energy requirement for the repair has a sigmoid type of behavior. There is a threshold, then there is a large increase, and then again there is a saturation. Similarly, the growth of cells also follows the same way, but there is a difference. There is a threshold, first of all, difference in threshold and difference in the slope, and the cell proliferation is linked with the misrepair and fixation of the damage. Now, both these repair and misrepair and fixation, they compete, and the result is either the cell survive after the damage or it is able to repair and again proliferate. So this is the work which was done on yeast, and then we verified this in HeLa cells and lymphocytes as a function, as a normal cell model, and finally also in the cancer patients. In the clinical trial, this was found that 2-deoxyglucose does not inhibit the next one, the damage in the normal cells. So further, you see the the depletion of ATP and the increase in ADP and AMP because of the lack of production of ATP, this leads to a number of reactions because the AMP activated protein kinase and mTOR, they result in reducing the expression of P53, which arrests the cell cycle at G1 checkpoint. And this is one of the major factors. Similarly, the signaling processes which are induced by the production of reactive oxygen species, they also create endoplasmic reticulum stress, ER stress, and also then encourage apoptosis and the autophagy. Next. So autophagy and the ER stress, they are also very important processes which determine the ultimate outcome, whether the cells will survive or die. Next. And this question of role of autophagy is very important because it has been shown recently that the ER stress or unfolding protein response are more important for autophagy rather than lowering of ATP levels. Now, autophagy is a very important process to create cellular resistance or alternatively cell death. If autophagy if ER stress continues for a long time and leading to autophagy, then it will make the cell sensitive, but if it is only for a short time, it may inhibit autophagy and the cell death. And therefore, one important point is that combining autophagy inhibitors with 2-deoxyglucose may be clinically useful. So this is an important point to remember. Now I come to the importance of this in vitro in silico study theoretical work, which was done by the molecular docking and molecular stimulations. So, as I told you earlier, the entry of coronavirus is dependent on the binding of this protein to cellular receptors, and this next. This is again in a diagrammatic form in a model system. So the entry is important, and the replication of the virus in the cell is important, which is both inhibited by 2-deoxyglucose. Important thing also is that the 2-DG showed no signs of active toxicity. In this theoretical work also this was studied. Next. Pointer. Now important study published in 1976 with the Japanese virus and in BHK cells. They showed that if 2-deoxyglucose is again replaced after a short time, after 24 hours, then the cells are able to carry out again the cell proliferation, they survive. So this is important, and that's why one has to schedule the 2-deoxyglucose in a proper way. Next. So this is in brief the mechanism, but mechanism is much more complex. We worked with the hypothesis that like the cancer cells, this 2-deoxyglucose will work also in virus-infected cells, and there was also some work done in Europe and also in Frankfurt University. They have started this work, but they have worked on the derivatives of 2-deoxyglucose. Similarly, the 2-deoxyglucose derivatives are also being tried in collaboration with the industry in USA. Now, what is our advantage? Our advantage is that 2-deoxyglucose itself has been tried here, and which is easily available and which has been approved by FDA also and by our ICMR and DGCI. And therefore, this 2-deoxyglucose could be obtained easily, and the permission of the government could also be obtained easily. And the clinical trials on COVID-19 patients could be tried by Dr. And in this, the very important role has been played by INMAS DRDO and Dr. Reddy's laboratory, because Dr. Reddy's laboratory got the patent transferred from DRDO to their laboratory in 2003. And they had all the know-how, how to manufacture, how to process these 2-deoxyglucose, which was developed by Dr. R.K. Sharma and colleagues in the DRD Gwalior, and they tested also its toxicity and purity. And after that, the patent obtained by DRDO was transferred to Dr. Reddy's laboratory. So they had the know-how and they could immediately produce and supply this 2-deoxyglucose. So next. So this is again summarizing the rationale and the conclusions from our studies. And next. Now, as you know, this 2-deoxyglucose use was announced about a week back, and it has been now, after the completion of the phase 3 clinical trials, it has now been released for emergency use. And there's a lot of information on the TV and on the press, among social media. So lots of enthusiasm and lots of hope that 2DG will be a game changer, and it's a very important development in India. But then lots of unanswered questions remain. Lots of unanswered questions remain, and whether 2DG can now damage the normal tissues? Can 2DG be given to all patients suffering from COVID? And what are the mechanisms? Now, these require further studies. So, in future directions, we we should optimize the efficacy, and combination of 2DG with other agents can provide synergistic action, and it can answer the question for reasons of resistance to treatment. You see, we do not expect that all patients, COVID patients, will be benefited by this 2DG. Some people, because there will be individual variability, some may not respond. But in order to answer these questions, we need to collect data on a large number of patients and also to carry out some basic work. And for this, a multidisciplinary collaborative research and various institutes must be carried out. We have some leads, for example, combination of 6CN, an inhibitor of pentose phosphate pathway, use of agents to increase the transport of glucose and glycolysis in COVID, and DNA binding agents like hooks to increase the differential effects of 2DG. These leads have been obtained from our work in INMAS mostly, and in National Institute of Mental Health in Bangalore. Next slide. Now I come to the second most important part of my presentation, that is integration of science and spirituality and behavior modification by yoga and medicine. And we have carried out some empirical studies on the effect of Pariksha meditation and dietary patterns. And these studies have been done on aggressiveness in school children, then some studies in adults and some studies on school teachers. These studies were in the school children was funded by a project of the Jain Vishwabharati Institute, and we are grateful to them for supporting this. And this work was then completed in 1917, and their comprehensive report was submitted to the university. And then we started studies on adults, and these studies on school teachers are going on. How much time? Please let me know the time available so that I can complete this within the time frame. Vandana?
Sir, it's an honor to listen to you, sir. You take your time, sir.
Okay.
Ji.
So just a few details about our results on this project in school children. Now, as you know, there are two types of aggressiveness. One is proactive and another is reactive. Proactive is induced by some goals, whereas reactive aggression is due to some response to previous injury, as a revenge kind of a thing. So both the proactive and reactive aggressiveness is coupled, and we have tried to study this in about 3,000 children, and this is the profile which we get. Now, important thing is that the children who have high proactive and high reactive aggressiveness, these are very few in number. It's around, let's say, a few percent, 3 to 4%. And these highly aggressive children they are prone to continue this aggressiveness in adult life also, and this creates disturbance because they are then prone to develop antisocial behavior and uh, juvenile delinquency and crime, committing crimes. This has been shown in many, many studies in all over the world, and therefore it is important to reduce this persistent high aggressiveness in these few school children. In America, the aggressiveness of school children is really a big problem because you always hear about the shootings by school children or and the government is trying to find out a solution. And they have tried a number of methods, including the cognitive behavior therapy, CBT, and they found some good results, encouraging results in huge clinical trials. But then it can't be applied on a large base, large scale, because it's too expensive and it's too time consuming. Because cognitive behavior therapy needs the help of a specialist clinical psychologist or psychiatrist and consultation with the community, in the community as well as in the school. So our method of using meditation is really very cost effective. And we have published these results in Indian Journal of Public Health and also in the Yoga International Journal of Yoga. And these are some of the results from these publications. So, so you see in the left part of the slide the profile, aggressiveness profile after Priksha meditation of 36 sessions. This shifts to this way. Now, important point is also that some small percentage of non-responders remain. And here it shows the as a function of time and or a function of sessions of this Priksha meditation schedule, modular, which we developed by the help of a late Swami Dharmanandji, who was at that time director of Adhyatma Sadhana Kendra in Delhi. And Dr. Pahuja, who was then the principal at the model school in Chhatarpur, and so he was very helpful, he was very enthusiastic, and so it could be started in the schools funded by South Delhi Municipal Corporation after getting their permission. So it's you see that already at around 18 sessions, and we conducted it on alternate days. So that means in a few weeks there is a tremendous amount of decrease, and there is after 36 sessions there is still some decrease, but not much. Next. Now about the non-responders. We tried to study this as biomarkers and biomarkers of autonomic nervous system and of HPA axis, which produces the cortisol, the stress hormone. The stress hormone was measured also non-invasively in the saliva of the children. And then we found, you see, these are the non-responsive, the dark blue. We found that there were significant differences, and therefore this uh, uh, combination of the biomarkers, that is measurement of COVID, measurement of cortisol, then measurement of the autonomic nervous system activity, the sympathetic nervous system, and the parasympathetic. The parasympathetic is the one which regulates and uh, uh, subsides the immediate response, is and therefore it's important. Now, what we see here is that the activity of the sympathetic system is actually decreased, is very low in these non-responders, whereas in these responders it's high. And similarly, the cortisol, the stress hormone. So these are very important indicators or predictors of the behavior of the children. Next. Now, I'll just come to the ongoing studies on the adults. This work was started with the help of Adhyatmik Sadhana Kendra in Delhi, and a number of psychological correlates of induced by perceived stress were studied, and among them, what is most important is to see the forgiveness and the quality of life in adults. The quality of life improves a lot if the forgiveness is enhanced and if the perceived stress is lowered. Next slide. So this just to show the effect of forgiveness, which reverses the effect of perceived stress and anger. So for peace, this forgiveness is very important for harmony. And therefore we wanted to measure this forgiveness. And it has been shown that with praksha meditation, forgiveness increases. So next. This is the relationship between anger and perceived stress. So anger is one response to the perceived stress, and this is the before praksha meditation, and this is after praksha meditation of 1 week practice in a group camp, in a shiver. And you see, though there is a lot of inter-individual variability, but on the average it is certainly anger is decreased. Next. So this also shows a relationship between the forgiveness and the quality of life. Next. Yeah, next. Okay. Now this work on forgiveness and the effects of reducing perceived stress and aggressiveness and anger in adults is going on, especially in teachers. But this work, which was being carried out by Mrs. Shweta Sharma, who is registered for PhD in JVBI, now could not be carried out during the lockdown period, that was the last 1 year. So this has been the problem, but whatever data she could gather, this is the result what I had shown you. So this we hope to be able to continue. So you see, the mental stress and mental problems and behavior of people can improve if they practice praksha meditation. this is the conclusion which we can draw, and this is supported by some of our ongoing empirical studies. So what I want to now say is that a combination of 2DG and with the spiritual technology of Priksha meditation can be very beneficial for the treatment of patients as well as for reducing the abnormal behavior in this public, because it has been noticed that the number of crimes and number of suicides in the last 1 year has increased drastically in India as well as in other countries in Europe or America. There are a number of studies which have shown this. So this is a very important aspect of this COVID-19 pandemic, the long-term effects on the socio-economic and the public behavior. And therefore, I think this is one of the very important result that practice of Priksha meditation can help in this. So a combination of 2DG and practice of Priksha meditation could be really very, very useful. And this must be continued in a large way, and we must develop multidisciplinary and multi-institutional collaborative programs in order to push it forward very rapidly. But, this is the message I want to convey, that international collaboration are very important between institutes of different disciplines, and the international collaborations are very important, which have played a very great role in developing these approaches. Next slide. In the end, I would like to acknowledge the help of so many institutes and so many people and funding agencies, mainly the ICMR, DRDO, DAE in India, in Germany, the GSF, German Academic Exchange Service, that is commonly known as DARD, and Alexander von Humboldt Foundation, and institutions, Goethe Institute, Frankfurt, where all the idea was developed in a simple model system of East, and then carried out in AIMS at different other model systems, and finally in the clinical trial, collaborations between AIMS, NIMHANS, INMAS, University of Essen, Patanjali Research Institute, and Savita University, and JVBI Lardmo. Next. And a number of people have been instrumental in coming and developing these techniques also to this stage. First of all, my mentors and teachers and students. Students are best teachers. This is my experience, and also is a proverb, is a popular experience. So Professor W. Polit, Professor R.K. Mishra, Professor A.K. Banerjee, Professor Ramalinga Swami, Professor P.N. Vahi, Professor Pentel, Dr. Arunachalam, and Dr. Abdul Kalam, the previous scientific advisors of DRDO, and Professor P.N. Srivastav, who was the Vice Chancellor of JNU and a radiation biologist himself, Professor Dr. Reddy, who was the director of NIMHANS, Professor Anta, and Mr. Nagratnam, and he was instrumental in recommending my name for the directorship of INMAS. Then the work on Priksha meditation Samni Professor Chetan Pragyaji, who was then the Vice Chancellor, who sanctioned the project for us. Then Samni Professor Chetan Pragyaji, who was ex-director of Bhagwan Mahavir International Research Center. Professor Stugar, who is the Vice Chancellor of JVBI, and Mr. K.C. Jain, who is the Director of Adhyatma Sadhana Kendra in New Delhi. And my students who are now, and colleagues, Professor V.K. Kalia, who is retired as a professor of biophysics at NIMHANS, Bangalore, Dr. B.S. Dwarkanath, who had joined me in INMAS after getting his PhD from NIMHANS, and he's now collaborating and with various institutes nationally and internationally. R.K. Sharma, who's Vice Chancellor of Savita, Dr. Khushu, who did a very remarkable work which I wanted to show you using the fMRI in studying the relationship between energy utilization by the brain and for carrying out various functions. He's now retired and is working in Bangalore as a consultant to various institutes and MRI. So Amitav Adhikari is now associate professor at the Oakland University in USA. Muruganandan also has migrated to USA. He's in the Ohio State University. Dr. Seema Gupta also migrated to US. Sudhir Chandna, who is now working in INMAS. Dr. Farad Khanam, retired from DRDO, the DFRL Institute in Mysore. Dr. Mrinalini Sharma, who has joined the Advanced Research Center at Indore, but now retired. And many scientists, Dr. A.N. Bhatt, Dr. Rajiv Varshne, Dr. Madhubala, Dr. Anil Mishra, Dr. Pallavi Thakur, Karan Jaina, who is now at the Apollo Hospital in Delhi, Professor B.K. Mohanty, he is Director of Cancer Institute in Odisha, Dr. Dinesh Singh, the now head of the radiation oncology in MAX, and Khushbu Jain, who is joined, I'm very happy, the Morarji Desai Institute, and Mrs. Shweta Sharma, Mr. Gautam Kumar, Mr. Ajay Sharma, who was working with me on the Kriksha meditation. Now, all these names are not all, a large number of people have really collaborated and contributed. I'm sorry that because of such a large number, I have not mentioned many of them. I ask for forgiveness if I have hurt anybody's sentiments, and I wish to thank all the people who have made this possible, and very grateful to Morarji Bhai Desai Institute for organizing this webinar, and to all the audiences and participants in this webinar. So thank you all for listening so patiently. And with this, I finish my talk. And open to questions if there are any. Yeah, thank you. आवाज नहीं आ रही.
Thank you, Professor Jain Saab. It was excellent. Thank you very much.
धन्यवाद.
Thank you, sir.
Yeah.
Questions will be taken by Vandana. Vandana, please take up the questions, please. Thank you.
Please. Sir, there are multiple questions on the social media platform, but I will take only 2 or 3 questions. So there's a question, has 2G been tried in vivo conditions or HeLa cells and primary cells? Does it not hamper the functioning of normal cells?
Yes, this we have shown in many systems, starting from yeast, where respiratory deficit mutant as a model for cancer and wild type as a model of normal cells. And this was then shown by Dr. Kalia in working at the time as AIMS in a whole body irradiation of mice and in HeLa cells. And this HeLa cell work was and lymphocyte work was carried out in collaboration with Cancer Research Institute of Tata Memorial Center in Mumbai and published, and one of the most cited papers. And also in the clinical trials, uh, Kaljena showed with the MRI images that in the normal brain tissue there was not any damage. Actually, there was less damage as compared to the controls. And similarly, Dr. Dwarkanath, together with some neurosurgeon, has published a paper on the brain tumor GBM patients and showed that the damage is reduced by 2-deoxyglucose. So I don't expect that there will be any damage in the healthy tissues of patients who are given to 2DG in COVID patients. But then this has to be seen in larger clinical trials, which are now possible because 2DG is becoming available. Yeah.
Sir, another question is, when we use 2DG as cells take glucose, 1 second, sir. When we use 2DG as cell take both glucose and 2DG, how the 2DG hampers cell proliferation?
Yeah, this I have tried to show that because of the greater reduction in the rate of ATP production and then greater effect of endoplasmic reticulum stress causing cell death and autophagy. This is producing this effect on cell growth. And therefore, virus-infected cells, they will not grow. Virus will not grow in these cells, and the cells themselves will not grow, proliferate. So that is how it works. Yeah. But this needs to be further investigated, and this is just the simple hypothesis on the basis of which we started this work. But the exact mechanisms are much more complicated. It affects also the immune system, and Dwarkanath and Dr. Bhatt have shown this in some model systems. So there are multiple actions, and we had shown that the gene expression in blastoma cell lines were changed in more than 100 genes. And this was dependent on the 2DG concentration and the time. And this work was done in collaboration with Wright State University in USA, where I had been after my retirement for some time. So this needs further to be investigated. What is the effect on the messenger RNA? What is the effect on the protein synthesis? But we know that this is affected by 2DG. How it is affected, this needs to be further understood, and more work on this needs to be done. And this can be done in collaboration with INMAS and AIMS and maybe NIMHANS. Thanks.
Okay, sir. So next question is, since virus multiplication is maximum in first week, shouldn't we give this 2DG in first week?
You see, if 2DG is able to inhibit the entry of virus also, then it is better to give it in the first week or even as a preventive. That means the general public, if it can take small amounts of 2DG in their diet or together with the diet, it may act as a preventive agent. But this needs to be investigated. But that's what I was saying, that it has lot of implications. But these implications have to be tested in vitro models and in animal models, and then also in the human patients. So a lot of data has to be collected. And now that 2-deoxyglucose will be used by a large number of patients in India, I think after a year or so, or when all the data is analyzed, we might get some clue that whether it can prevent. And if it can prevent, then it will be extremely useful because it's not specific to COVID-19. All enveloped viruses can be prevented, and therefore in future, if there are any pandemics, then they can be prevented by this 2-deoxyglucose. And also in our hospitals, people who are in the ICU, they usually develop hospital infections, viral infections, which are deadly. Now, even in if it is tried, even in those hospital infections, it could be useful. So it has many, many uses. And this clinical trial with COVID-19 can work as a agent to induce all these, to initiate all these various uses also. So that's why the contribution of INMAS, DRDO, and Dr. Reddy's lab is very, very important for future developments also. And we must congratulate them. And we must encourage a multidisciplinary, multi-institutional trial on emission mode in India. Then only it will gain also recognition from the world. So all the underdeveloped countries will be benefited because it's a very simple and cost-effective application, and it can be done even at home. So, but all these things have to be verified in the long-term clinical trials. Large number of data has to be collected. Thank you.
Sir, is your permission to take one question?
Yeah, please.
So somebody has asked 2D glucose publicly कब तक available होगी?
No, I'm not in a position to answer that. That only Dr. Reddy's lab can provide the answer. But I think I have been told they are working hard on it. But somehow or the other, it is delayed because they started the work at least last year. So it's more than a year that they have been at it. And why there is a difficulty, I don't know. But INMAS people or Dr. Reddy's lab people can answer that question better. Yeah.
Okay, sir. Sir, on behalf of Morarji Desai National Institute of Yoga and our social media friends, I extend my heartfelt gratitude to you, sir, for delivering such an extreme informative lecture. Thank you so much for coming to us today. So it's an extreme pleasure to hear from you, sir. Thank you so much. I would also like to thank Director of MDNIY, Dr. IV Basavaraddi sir, for his vision and support, and our social media friends, CNDO of MDNIY, and all our viewers for joining us today. Now let us end the program with Shanti. सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामया सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् मा कश्चित् दुख भावे ओम शांति शांति शांति. Thank you so much, sir.
Namaskar.
Thank you. Thank you, sir. Thank you, Professor.
Satyam Sundaram.
Thank you.
Satyam Sundaram.
Thank you. Thank you.
Thank you.
It's a great pleasure. Really, it was very, very informative and inspiring, sir. Thank you.
धन्यवाद. चलो.